Hello everyone, welcome to Science Nerd. Today we're going to talk about protein folding. So whenever we are studying any topic of science, we need to answer three questions. Why, how and when. So our first question is why? Why are proteins folded? Or why is it important for us or for the cell to fold the protein? So it says that a protein is made up of amino acid that you all know by now. So proteins in their native structure are only functional. In order to make the protein functional, they have to be in their native structure. And what is the native structure of the protein? It is the 3D structure. Is their native structure. And answer for why is to attain function. Right? So the second question is when? When are proteins folded? Proteins are folded just after their synthesis. When proteins are biosynthesized, just after or sometimes even during their biosynthesis, proteins are folded. So this is a fast reaction. So during or after biosynthesis. So the third question is how are proteins folded? What assists protein folding? Like what helps the proteins to fold? So the answer is temperature. Protein folding depends on temperature. Second is solvent. Third is salt concentration. And fourth is there are some molecules, there are important molecules that help in protein folding. What are these molecules? These molecules are called as chaperons. So, why? Because to attain 3D structure or their native structure for their functionality when, during or after biosynthesis, and how? By temperature, by solvent, by salt concentration and by chaperons. So I think this is clear to you. Moving on. There was an experiment done by a scientist in the early 1960s. The scientist was named Christian. So he did an experiment. He took a protein molecule, a completely folded protein molecule. He added what kind of reagent? Denaturating agents. क्या होते हैं denaturating agents? ये ऐसे agents होते हैं. These are the agents that break down something, denature their conformation or break down their conformation. So the two reagents he used was urea and beta mercaptoethanol. So he used urea and beta mercaptoethanol. So obviously when denaturating agents were used, the protein conformation would denature and they would find their primary or secondary structure. 3D so they would change into 2D. Correct? Easy to clear? So what happened was, but when he removed these denaturating agents, okay, again the proteins, they came back to their original native state. They came back to their original 3D conformation. Why was it so? Because maybe amino acids, they interact via hydrophobic bonds or hydrophobic interaction to form back to their original state. But the second question is that if they come back to their original state, is there a chance of some kind of mistake that protein folding might occur while the proteins are folding? Yes, obviously, because there are only 20 amino acids. When they are joining together, there may be some kind of mistakes. So, in order to reduce the mistake, we have a molecule called as chaperons. So, from protein folding, we come to an important topic called chaperons. Chaperons, they help in protein folding. First function of chaperons is they help in protein folding. Second is, if the unfolded or incorrectly folded proteins are formed, chaperons may help them to go back to the original or to the correct protein folded form. They may even help other proteins to translocate them. So, second is translocation of other proteins. Third is aggregation of proteins. When proteins are not folded correctly, they may aggregate. Aggregation means they may clump together. वो इकट्ठे हो जाते हैं. Aggregation is bad. So chaperons, they may prevent aggregation of proteins. And the fourth function is when the proteins are in incorrect folding or not folded properly, they may form into prions. Prions cause diseases, right? Disease in animals and humans, guru disease, the mad cow disease. So, chaperons, they prevent prion formation. So these are the functions of chaperons. Moving on, what basically are chaperons? These were the functions, like where chaperons are used. But what basically are they and where are they found? They are ubiquitous. Ubiquitous का मतलब found everywhere. Found everywhere, that is, they are found in prokaryotes, eukaryotes and archaea. That is in every type of cell. So, chaperons, they may be classified as HSPs. HSP is heat shock protein. Why were they called HSP? They were called HSP because when they were found in cells which had a slightly higher temperature. That is, the normal temperature of a cell is 37 degrees, but they were found when the temperature was around 42 degrees Celsius. So that is why they were called heat shock proteins. So examples of HSP, we have heat shock protein 40, heat shock protein 60, heat shock protein 70, heat shock protein 90, heat shock protein 100, and etc. So different classes of heat shock proteins are found in different types of protein folding. So the two main families of heat shock protein or the HSP chaperons are the HSP 70 and the HSP 60. So these are the two classes of heat shock proteins or the two families of heat shock protein. So the HSP 70, they are found early in protein life, and the HSP 60, they are found late in protein life. That is after synthesis of protein. After the synthesis of protein, the HSP70, where is it found? It is found on string of around 7 amino acid. There is a string of 7 amino acid forming a protein. There it is found. And the amino acids are hydrophobic on a string of around 7 hydrophobic amino acids. And where is the HSP60 found? They are found in the HSP60 unfolded, partially folded, or incorrectly folded amino acids, or I should say proteins, not amino acids. When there is an unfolded protein or an incorrectly folded or partially folded protein means a molten globule. I'll tell you about it again. Okay, so here heat shock protein 60 are formed. Also, a thing about heat shock protein 60, they are called chaperonins. The HSP70, they have 2 subunits, or I should say 2 functional subunits. One is that binds and hydrolyzes ATP, that helps in hydrolysis of ATP, and the second is that binds to unfolded proteins. And in the HSP60, the HSP60 family or the chaperonins, they have 14 subunit bound in a heptameric form. That is 7 like this and 7 like this in inverse form. Like this. They form a cavity. And what happens in this cavity? In this cavity, the unfolded protein, this goes, and then ATP joins, there is a thing as called as co-chaperonin present. It then correctly folds the protein. Then the co-chaperonin removed. This cap is removed. This is the cap, it is removed, and the protein, the correctly folded protein comes out. So this, the heptameric subunits are called GROEL in case of bacteria and HSP 60 in case of eukaryotic cell. And the co-chaperonin, it is called GROES in case of bacteria and HSP10 in case of eukaryotic cell wall. So this is all about chaperons. So when this GROEL or HSP60 it becomes free, again an unfolded protein can come into this cavity, again ATP comes, again co-chaperonin, that is GROES, and HSP10 comes, caps it, and then the protein becomes completely folded, again it gets removed. So that is how it works. So the protein folding steps in protein folding, and one intermediate step is, or one intermediate is formed. And what is that intermediate? This is your not folded protein. There is a fast reaction forming a molten globule. Which is partially folded structure. It is not a completely folded protein. And the molten globule, it forms into a native structure. So this is all about protein folding, how proteins fold, why is it important, when it happens, and what chaperones are, how do they function. This is all about this, and I hope you liked the video. For the next class, we may talk about protein structures, the primary, the secondary, the tertiary, and the quaternary in detail. That will be a playlist that will come in series, and I hope you like the video. Do subscribe to my channel and like, share and subscribe. Thank you.